Bismillahirrahmanirrahim. In the name of Allah, the most merciful and the most beneficent. Assalamu alaikum. My name is Ifali and I am student of PSY Technology from University of Management and Technology. The title of my topic is Nobel Prize winner of 2022 in the field of medicine and philosophy. I hope you find this video quite helpful. Svantee Paabo was a scientist who pioneered the field of evolutionary genetics. Actually, he was the one who studied early humans and Asian population. I myself feel quite intimidated to explain his beautiful research. The topics which I am going to discuss are introduction, research, education, DNA sequencing, evolutionary genetics, evolution, genetics, Hardy-Wingberg theorem, and at last I am going to conclude my topic. Svantee Paabo was born in 1955. Svantee Paabo, Swedish evolutionary geneticist who specialized in the study of DNA from Asian specimens and who was the first to contribute to the sequencing of Neanderthal genome. Neanderthal genome is the genome of extant organisms. Paabo also discovered the hominian denivosa. For his research, he was awarded Nobel Prize in the field of evolutionary genetics. The 2022 Nobel Prize for Philosophy or Medicine has been awarded to Swedish geneticist Svantee Paabo for his research in the field of genomes of extant hominians and human evolution. Now Background of Svantee Paabo. Paabo's parents were scientists, his mother being a chemist and his father biochemist Sunke Bergström. Having won a share of 1982 Nobel Prize for Philosophy or Medicine, Paabo also pursued his own career in the sciences. Paabo wrote in his 2014 book Neanderthal Man, In Search of Lost Genomes, that he is bisexual. He assumed he was a guy until he met Linda Vigilant, an American primatologist and genetic education of Svantee Paabo. He was a student of School of Interpreters, Swedish Defense Forces in 1975 to 1976. In 1975 to 1981, he studied at the Faculty of Humanities, University of Uppsala, including the history of science, Egyptology, and Russian. In 1977 to 1980, medical studies at University of Uppsala, Sweden. In 1979 to 1980, part-time research and teaching at the Department of Cell Biology, Uppsala, and Ruge Institute for Molecular Biology, Nutle, NG, USA. In 1981, full-time research as PhD student at the Department of Cell Research of Uppsala. In 1986, PhD degree at University of Uppsala, Sweden. In 1986 to 1987, postdoctoral research at the Institute for Molecular Biology to University of Zurich, Switzerland. In 1987, short period of work at Empirical Cancer Research Fund, London, UK. In 1987 to 1990, postdoctoral research at Department of Biochemistry, University of California at Berkeley, USA. In 1990 to 1998, full professor of general biology, University of Munich, Germany. In 1997, director of Institute of Max Planck for Evolutionary Anthropology, Leipzig, Germany. In 1999, professor of genetics and evolutionary biology, University of Leipzig, Germany. In 2003 to 2015, guest professor of comparative genomics, University of Uppsala, Sweden. In 2016, honorary research fellow, Natural History Museum, London, UK. In 2020, adjunct professor, Okinawa Institute of Science and Technology, Japan. Next is profession of Svantee Paabo. Dr. Paabo was appointed as a professor at the University of Munich in 1990, where he continued his research to study DNA from extant human species. This was when he decided to study mitochondrial DNA from Neanderthals. Mitochondria, popularly called the powerhouse of the cell, is an organelle inside the cell that has its own DNA. That is why it is also called as self-replicating organelle. Although the mitochondrial genome is small and only contains a fraction of genetic information in the cell, It is present in thousands of copies. This increases the chance of its successful sequence. Research and career of Svantee Paabo. Paabo is known as one of the founders of paleogenetics, a discipline that uses genetics to study early humans and other Asian species. From 1986 to 1987, he did postdoctoral research at Institute for Molecular Biology to University of Zurich, Switzerland. Research in 1987 to 1997. As an AMBO postdoctoral fellow, Paabo moved to the United States in 1987, accepting a position as a postdoctoral researcher in biochemistry at the University of California, Berkeley, where he joined Alan Wilson's lab and worked on the genome of extant mammals. In 1990, he returned to Europe to become professor of general biology at University of Munich, and in 1997, he became founding director of Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. Research in 1997 to 2002. In 1997, Paabo and colleagues reported their successful sequencing of Neanderthal mitochondrial DNA originating from a specimen found in Feldford Grotto in the Neander Valley. In August 2002, Paabo's department published findings about the language gene FOXP2, which is mutated in some individuals with language disabilities. Research in 2006 to 2009. In 2006, Paabo announced a plan to reconstruct the entire genome of Neanderthals. In 2007, he was named one of Time magazine's 100 most influential people of the year. In February 2009, at the annual meeting of American Association for the Advancement of Science in Chicago, it was announced that the Max Planck Institute for Evolutionary Anthropology had completed the first draft version of Neanderthal genome. Over 3 base pairs were sequenced in collaboration with the 454 Life Sciences Corporation. In order to gain a better understanding, let us look at this video. This shows that mutations create a lot of differences and how they vary from each other. The colored nucleotides show the mutation. This is how we are different from each other and how humans evolved from each other. Next is research in 2010. In 2010, he published the first Neanderthal genome sequence. DNA sequences from Neanderthals were also found to be more similar to sequences from contemporary humans originating from Europe to Asia than to contemporary humans originating from Africa, suggesting interbreeding between Neanderthals and Homo sapiens. Research in 2010 to 2014. Paabo doctoral student Vivian Salon was able to successfully map the Denisovan genome, clarifying the geographic distribution and admixtures in Arctic humans. In 2014, he published the book Neanderthal Man in Search of Lost Genomes, where he, in the mixed form of memoir and popular science, tells the story of a research effort to map the Neanderthal genome combined with his thoughts on human evolution. Research in 2020 to 2022. In 2020, Paabo determined that more severe impacts upon victims of COVID-19 disease, including the vulnerability to it and the incidence of the necessity of hospitalization, have been associated via DNA analysis to be expressed in genetic variants at chromosomal region 3. Features that are associated with European Neanderthal heritage. That structure imposes greater risk that those affected will develop a more severe form of the disease. The findings are from Paabo and researchers he held at the Max Planck Institute and Karolinska Institute. In order to gain a better understanding, let us look at this video. This is a cell which shows the composition. It has a nucleus at the center and mitochondria, which is also called powerhouse of cell. We have separated the DNA of mitochondria and now we are studying its detail. As you can see, there are 16,500 base pairs, out of which 3 million base pairs are present in our DNA. And the red color dots show the mutation, how they vary from other humans. Svantee Paabo work. His first work in paleogenetics on sequence from Egyptian mummy was the world's second successful one and posed proof that nuclear DNA can survive from ancient times in a state viable for further analysis. Paabo further worked range from research on sequence from other ancient samples, for example, 7,000-year-old brain of an ancient Native American excavated from a blog in Florida to ones focusing on exploring and appraising methodology currently available in molecular biology. For example, a publication comparing results obtained through molecular cloning and PCR, the first publication recording genetic material from ancient hominids, appeared in 1998 when Paabo's team reported recovering mitochondrial DNA from Neanderthals. Hope you guys understand better by looking at this picture of 7,000 years old brain of American native. Success of Svantee Paabo work. The full mitochondrial genome was sequenced in 2008, while in 2010 appeared the first draft of Neanderthal genome, followed by high coverage Neanderthal genome in 2014. The obtained data have finally provided proof for the admixture of Neanderthal DNA to the modern human genome, which happened fairly recently, 70,000 years ago and long after the populations diverged. Importance of Svantee's research. Thanks to Svantee Paabo discoveries, we now understand that archaic gene sequences from other Asian relatives influence the philosophy of present-day humans. One such example is the Denisovan version of the gene EPAS1, which confers an advantage for survival at high altitude and is common among present Tibetans. Of Svantee research. Paabo's research has resulted in the rise of a new scientific discipline called paleogenomics, which is the study and analysis of genes of ancient or extant organisms. The prize was announced on Monday by the Royal Swedish Academy of Sciences in Stockholm. Genomics. It is defined as study and analysis of genes of organisms that are ancient and extant. Ancient human bone with DNA from various species, then it is inserted to NGS library, then it is divided into three parts: shotgun, nuclear DNA capture and SNP capture, where we can find the read ratio in DNA capture. This shows the different composition of bacteria, viruses, fungi, animal, human in each organism. Opinion of other scientists about Svantee Paabo work. Partha Majumdar, National Science Chair of Science Engineering Board, Government of India, not only praised Dr. Paabo's research but also the technological aspect of it. His conceptual breakthrough is of paramount importance in understanding human evolution, but at the same time his technological breakthrough deserves equal praise. It is not easy to amplify and sequence ancient DNA because it is highly fragmented and full of contamination from microorganisms like fungi and bacteria. Dr. Majumdar said to the Hindus, "It remains to be seen if the renewed interest in the field will lead to better funding and subsequently more opportunities for researchers." This is what Dr. Majumdar added in his development on Svantee Paabo work within this year. The scientist also said that the Nobel laureate's research has helped in furthering the recognition of evolutionary biology and paleogenomics. This is a hard field to follow, especially in places like India and Africa, because the Asian DNA is not preserved well in tropical weather conditions. Chimpanzees. Great apes, gorilla, chimpanzee, bonobo and human resulted in the inference that the human line of descent split from the chimpanzee line about 5 million years ago. Chimpanzees are therefore our nearest non-human relative. DNA changes accumulate over time and create diversity in a group of individuals. Thus, it is logical to conclude that a group of humans with a highly diverse set of DNA sequences is older than a group whose sequences are less diverse. Let's have a better understanding about this by looking at this video. As you can see, we are somehow evolved from monkeys, as we can see from their bones, but the difference is their bones are somehow replaced by the positions. As we can walk like monkeys. But this is the species that have been extant, and Svantee Paabo aim was to determine the genome of extant organisms. Hofer Cave. This is when Paabo's group encountered some major scientific challenges. Bones that lie around for many thousands of years are exposed to the vagaries of nature. Microbes, notably bacteria and fungi, grow on such debris. These microorganisms also have DNA. Contaminating DNA. Contaminating DNA would be a tiny fraction of total DNA extracted from the bones and thus does not matter. However, the quantity of DNA in a sample is usually too small for its sequence to be determined, and it has to be amplified by a process called polymerase chain reaction. Kerem Muesli was awarded a Nobel Prize in Chemistry in 1993 for his role in the invention of PCR technology, without which most DNA research undertaken today would not have been possible. This shows that chemistry is everywhere, and chemistry is quite important, and each and every field needs chemistry in order to gain a better understanding. The scientific fields are somehow always related to each other, and we have to know them in order to know their importance. And in order to know the importance of chemistry, I am just suggesting or sharing this beautiful video with you guys. Hope you now understand the importance of chemistry at the same time with the field of biology, genetics and evolution. Challenge during DNA extraction. Some challenges observed during DNA extractions are as follows. The bones exposed to environment, DNA degradation or fragmentation or cross-linking or deamination take place. This degradation may be too much harmful that the extracted DNA may be unsuitable for analysis. However, undergoing through these challenges, Paabo was the scientist who extracted DNA from ancient bones. Neanderthal DNA in modern humans. Statistical analysis of catalog of DNA variants of Neanderthals and humans allowed Paabo to conclude that Neanderthals and humans did. Neanderthals contributed about 1 to 4% to the DNA of non-Africans. One study claimed this contribution to be 20%. DNA analysis of small bones found in Denisova cave in Siberia revealed that the individuals from whom these bones came were different. DNA extraction from two different species. In 2010, Neanderthals and the two Denisovans showed that Neanderthals and Denisovans had also interbreed, and that Denisovans contributed 4 to 6% to non-African DNA. In 2018, DNA extracted from another bone found in a Denisovan cave and identified that bone as belonging to a child of Neanderthal father and Denisovan mother. This is the picture during the research. Karolinska Institute. If modern humans interbreed with Neanderthals and Denisovans and produced children, why did these species become extinct? The likely reason is that the children of the unions between humans and Neanderthals and Denisovans remained with the humans over thousands of years. The population size of hominian species started to shrink, and they became extinct. When humans and Neanderthals interbreed, children who received from Neanderthals the immune genes with the variants that conferred resistance had a better chance of survival than those who did not. Such resistant variants obtained from Neanderthals have been found among Europeans but not among Africans. As you can see from this picture, Denisovans have a more hairy structure than the Neanderthals, and that is why Neanderthals' genomes is far better than Denisova. Let us know what is DNA. DNA, abbreviation of deoxyribonucleic acid, organic chemical of complex molecular structure found in all eukaryotic and eukaryotic cells and in many viruses. DNA codes genetic information for the transmission of inherited DNA. This shows from single DNA strand. 2 DNA strands can be formed by using each strand as a template, and the new strand also produced. DNA structure is shown. It is made up of base pairs, phosphate and sugar. The sugar used is deoxyribosugars, and the backbone is made up of phosphate backbone, whereas the nucleotides make combination are adenine form bond with thymine. There is double bond, and cytosine form triple bond with guanine. Pyrimidines are cytosine and thymine, whereas purines, which are double bonded rings, are adenine and guanine. Of DNA. The chemical DNA was first discovered in 1869, but its role in genetic inheritance was not demonstrated until 1943. In 1953, James Watson and Frederick Crick, aided by the work of biophysicists Rosalind Franklin and Maurice Wilkins, determined that the structure of DNA is double helix polymer, a spiral consisting of two DNA strands wound around each other. The breakthrough led to significant advances in scientists' understanding of DNA replication and heredity control of cellular activity. The structure of DNA bases, which are adenine, guanine, cytosine, thymine. Thymine and adenine form double bond and they are the combination of purine and pyrimidine bases, whereas guanine and cytosine form triple bond, which is the combination of purine and pyrimidine. DNA sequencing. DNA sequencing refers to a general laboratory technique for determining the exact sequence of nucleotide or bases in a DNA molecule. The sequence of bases encodes the biological information that cell uses to develop and operate. Establishing the sequence of DNA is key to understanding the function of gene and other parts of the genome. In order to gain better understanding of DNA sequence, let us look at this picture. The first strand is shown. This shows that cytosine always form bond with guanine and thymine always form bond with adenine. Then this is shown that the first strand go to the sequence of the template of the strand. It is larger fragments always move downward towards smaller fragments. Observation of DNA sequence. The first DNA sequence were obtained in the early 1970s by the academic researcher using laborious methods based on two-dimensional chromatography. Following the development of fluorescence-based sequencing method with the DNA sequencer, DNA sequencing has become easier and orders of magnitude faster. Sanger sequencing. In Sanger sequencing, the target DNA is copied many times, making fragments of different lengths. Fluorescent chain-terminated nucleotides mark the ends of the fragments and allow the sequence to be determined. Next-generation sequencing techniques are new, large-scale approaches that increases the speed and reduce the cost of DNA sequencing. Methods to extract DNA. Genetics has made the most robust contribution to this understanding, the invention of methods to extract DNA from the cell and to read its linear sequence. Nucleotide by nucleotide. DNA alphabets are also called nucleotides has made this possible. Steps for to extract DNA is first the cell is broken down. It is so that the DNA is extracted. Then the DNA is resuspended so that we can study the sequence of nucleotides. Difference in nucleotide sequence. DNA of many individuals is over 99.9% similar. Human DNA has about 3 billion nucleotides, so a difference of 0.1% is a difference of about 3 million nucleotides. This percentage of DNA is possible for uniqueness of humans. DNA sequencing is also obtained or extracted by these methods. First is organic extraction, through which the nucleotide sequence from DNA is used. Next it is Charles extraction. Same case, the nucleotide sequence from DNA is also extracted through this process. Third is solid phase extraction, in which we use magnet to separate the DNA molecules so that we can determine the nucleotide sequence. In order to understand better, let us look at this video. This shows that there are in actual 3.2 billion base pairs, out of which 3 million base pairs create differences. The yellow color or maroon color shows the differences or the mutations in which the organisms vary from each other. The same, if we have the same nucleotide sequence, then we cannot evolve from each other. We will have the same traits. The last and the most important question: where did we come from? Different religions have different answers. Science has a different answer. Charles Darwin proposed with reasonable observational support that humans arrived by gradual biological changes from apes-like ancestors. Thus, the belief that humans were created was dispensed with over the years. Darwin's proposal gained strength, especially after scientists were able to analyze DNA. Genetics. Evolutionary genetics is a combination of two words, evolution and genetics, which shows that how genetic variation led to evolutionary changes. Charles Darwin and evolution. It is not the strongest of the species that survive, nor the most intelligent, but the one most responsive to change. This shows the importance of change. As we have heard, nothing is permanent in the world except change. If we want to adjust in a new environment, we have to adopt changes according to the environment. Evolutionary genetics. Nothing in biology makes sense except in the light of genetics. This is a very important and a very beautiful line. Importance of evolutionary genetics. Evolutionary genetics include population genetics uses both molecular and classical genetic methods to understand the organic and effects of mutation and recombination. It applies empirical studies and theoretical methods to uncover the roles of genetic drift, gene flow, natural selection to predict and interpret evolutionary change. Perhaps the most important finding in this field is the large amount of genetic variation in protein phenotypes that seem to have no immediate selective value. Furthermore, recombinant DNA technology has revealed that there are large amounts of previously undetected polymorphism at the nucleotide level that do not affect change in the nucleic amino acid sequence. Consistency in protein sequence in the face of nucleotide polymorphism shows that selection is acting directly at the DNA level, preventing the evolution of deleterious proteins. Evolutionary genetics. Neo-Darwinism is also called as modern evolutionary synthesis. Generally denote the integration of Charles Darwin theory of evolution by natural selection. Gregor-Mendel theory of genetics as the basis for biological inheritance and the mathematical population genetics. Although this was not the historical meaning of the term Neo-Darwinism, it has been the popular and scientific use of the expression since the synthesis of the 1930s. The modern synthetic theory of evolution. Initial start was in 1940. Sources of variation were mutation, recombination, crossing over, independent assortment, and agents that produces changes were natural selection, drift, mutation, migration, non-random mating. Traditional view was proximate change. Phenotypes were produced by two ways. Non-evolutionary science says the mechanism of development that you acquired characteristic or according to your strength, according to your hard work, according to your struggle. Second was the phenotype developed by natural selection and genetic drift. This is called evolutionary science. This is the map which shows somehow the changes, the evolution of different species in different countries. Role of evolutionary genetics. Evolutionary genetics include population genetics uses both molecular and classical genetic methods to understand the origin and effects of mutation and recombination. It employs empirical studies and theoretical methods to uncover the role of genetic drift, gene flow, natural selection to predict and interpret evolutionary changes. Evolutionary genetic forces. There are three main forces for evolutionary genetics. First is genetic drift, second is selection, third is mutation. Genetic drift. Genetic drift is an evolutionary change in allelic frequency of a population as a matter of chance. It occurs in very small population, but its effects are strong. It occurs due to an error in selecting the alleles for the next generation from the gene pool of the current generation. It does not occur due to any environmental influences. In large population, the allele frequency of the gene remains relatively stable because the genes are not affecting fitness and do not have a natural selection pressure against the alleles. Genetic mutation is shown. As you can see the difference in original sequence, and first is delegation, means that a nucleotide is removed. Second is original sequence and insertion, in which an extra nucleotide is added. Third one is original sequence and point mutation, in which we have find a different nucleotide as compared to the previous one. This is genetic drift. Parent population with significant genetic variation, then post-bottleneck population where all but the three yellow-green alleles have been lost. Mutation. Mutation is a change in the DNA sequence of an organism. Mutation can result from errors in DNA replication during cell division, exposure to mutagens, or a viral infection. Germline mutation occur in eggs and sperms can be passed on to the offsprings, where somatic mutations that occur in different body cells cannot pass on to the generations. Selection. The theory of evolution by natural selection was proposed by Charles Darwin and Alfred Russel Wallace in 1858. They argued that species with useful adaptations to the environment are more likely to survive and produce progeny than are those with less useful conditions, thereby increasing the frequency with which useful adaptations occur over the generations. This is what is natural selection. Natural selection refers to those organisms that are able to survive in that environment with their traits or favorable characteristics. Conclusion. In short, I'm going to conclude my topic with these few lines. These are not enough to conclude my topic, but I have to summarize my topic with these important short points. Actually, Svantee Paabo determined the DNA sequence of ancient organisms from their fossils. Evolutionary genetics have played an important role in our daily life. It enhanced the study of other fields of science and played an important role in modern technology. Criminology is decreased by the advancement in genetics. Relationship issues have been. The of natural selection is long-necked giraffes. What is the advantage of long-necked giraffe? Giraffe uses their long neck to eat leaves from the top of the trees. Male giraffe uses their long necks to fight with other male giraffes for access to females. Giraffes are also able to see predators from greater distance away. Evolution. Evolution is a process that results in changes in the genetic material of a population over time. Evolution reflects the adaptations of organisms to their changing environment and can result in altered genes, novel traits, and new species. Evolution processes depend on both changes in genetic variability and changes in allelic frequency over time. 1744 to 1829. Jean-Baptiste Pierre-Antoine de Monod, often known as Lamarck, was a French naturalist, biologist, academic, and soldier. He was an early proponent of the idea that biological evolution occurred and proceeded in accordance with the natural laws. Theory of evolution according to Lamarck. According to Lamarck, there are 2 points that represent theory of evolution: use and misuse of organs and acquired inheritance. Original shock-led ancestors keep stretching neck to reach the leaves higher up on the tree, and stretching until neck become progressively longer. Use and misuse of organ. The inheritance of acquired characteristics means its reappearance in one or more individuals in the next or in succeeding generations. An example would be found in that supposed inheritance of a change brought about by the use and misuse of special organ. Example, blacksmith arm. Lamarck argued that a blacksmith gets strong muscles from his work and his son inherited those strong muscles. However, his son not inherited those strong muscles. He struggled and worked hard for those strong muscles. Criticism of Lamarckism. There are 2 main points that explain the criticism of Lamarckism. Though the tendency to increase in size has been shown in many forms, there are also examples where there is reduction in size. For example, trees that are primitive are large in size, while the shrubs, herbs, grasses that evolve later are smaller in size. If new organs were to develop in response to a new need, that man should have developed wings by now. Factors that justify Lamarckism. The factors that justify are as follows. Effect of environment. For example, radish plant grows in 2 years in cold weather while in 1 year in tropical weather. Effects of chemical. Change in amount of hormones affect different parts of the body. For example, if somatostatin hormone is produced in large amount, then it leads to greater growth of human body. Regeneration of parts like starfish. Starfish, if cut off his arm, it can regenerate through its neurons or receptors. Darwin, 1809. Charles Darwin, in full name Charles Robert Darwin, England, died in April 19 in 1882. The English naturalist whose scientific theory of evolution by natural selection became the foundation of modern evolutionary studies. Theory of evolution. Charles Darwin's theory of evolution had 3 main components. Variation occurred randomly among the members of a species, and individual traits could be inherited by its progeny. The struggle for existence would allow only those with favorable traits to survive. The example or the picture shows the things we have already discussed before. Now, the parts of natural selection or the whole components or factors of Darwin's theory of evolution are as follows. First one is genetic drift. Random stochastic processes changes the allelic frequency. Second is natural selection, where differences in productive success of alleles changes allelic frequency. Third is recombination, new combination of alleles, no change in allelic frequency. Fourth, mutation, new allele formed, change in allelic frequency. Migration, new alleles introduced, change in allelic frequency. Sixth one is population growth, expansion of population size, no change in allelic frequencies. Factors that justify Darwin theory. There are several factors that justify Darwin theory of natural selection. First one is overproduction, selection, adaptation, speciation. Adaptability. This shows that the organisms which adopt itself according to the environment is able to reproduce and survive in this competitive environment, and this is referred to as survival of the fittest. The reason behind this is because of the competitive environment, short resources, and a very large population. Survival of the fittest refers to the natural process resulting in the evolution of organisms that make them acquire a better design that is to fit to the environment. Second is variation. Genetic variation can arise from genetic variants, mutation, or from a normal process in which genetic material is rearranged as a cell in getting ready to divide. Genetic variation that alter gene activity or protein function can introduce different traits in an organism. If a trait is advantageous and helps the individual survive and reproduce, the genetic variation is more likely to be passed to the next generation. As you can see, the original correct copy and the original and mutant copy, there is difference in the nucleotide sequence. Overproduction. Overproduction in actual leads to increase in population, which in actual leads to competition, which result in shortage of resources. Overproduction led to a lot of problems. For example, overproduction caused violation and many other problems, just like crime increases, the rate of crime increases a lot, that people start robbing others just to complete their necessities. Genetics. Genetics is the study of genes, genetic variation, heredity in organisms. It is an important branch in biology because heredity is vital to organisms' evolution. Genetics is the study of genes, genetic variation, and heredity in organisms. It is an important branch in biology because heredity is vital to organisms' evolution. Evolutionary genetics, Mendel and genetics. Krieger-Mendel experiments were performed in 1856 to 1865. His work remained unappreciated or unpraised until 3400s after his death. Mendel's rediscovery and early modern geneticists were De Vries, Chester Much, Coran, who in actual appreciated Mendel's work into 1900s. Genes, phenotypes, population dynamics, community structure, ecosystem function are somehow always related to each other. Neo-Darwinism. The modern synthesis of evolution is based on the following ideas and makes significant modification to Darwin's view in its understanding of causes contributing to evolution to width. Population contains genetic variation that arises by random, not adaptability, mutation, and recombination. Population evolves changes in gene frequency brought about by random genetic drift, gene flow, and especially natural selection. Modern synthesis, Neo-Darwinism is genetic mutation, Mendelian inheritance, which is only related to DNA inheritance, population genetics, and contingency is related to gene selection, and speciation and transgenome isolated. Darwinism is variation, inheritance, and natural selection, whereas integrated synthesis is evo-devo theory, plasticity and accommodation, epigenetic inheritance, multi-level selection, genomic evolution, niche construction, replicator theory, and volubility. Minimum theorem. In a large random mating population, the genotype allele frequency remain constant in the absence of any evolutionary influence from one to another generation. Influences are inclusive of a choice of mate, natural selection, genetic drift, mutation, sexual selection, gene flow, genetic hitchhiking, founder effect, meiotic drive, population bottleneck, inbreeding, and associative mating. Proposal of Hardy-Wingman theorem. The law is named after D. H. Hardy and William Windberg. They were pioneers in mathematically illustrating this principle, often referred to as Hardy-Wingman theorem. Concluding, I have to conclude my topic with these few lines. However, they are not enough to explain the whole topic, but I have to summarize my topic with these important points. Actually, Svantee Paabo determined the DNA sequence of ancient organisms from their fossils. Evolution genetics have played an important role in our daily life. It enhanced the study of other fields of science and played an important role in modern technology. Now, criminology is decreased by the advancement in genetics. Relationship issues have been declined due to study of genetics. Svantee Paabo showed his interest in hominids and studied the immunity traits gene in them. These are the links from where I have concluded the whole topic or research of all my topic belong to these links. Thank you so much. I hope you guys like this video and I hope you may find this video quite helpful. In short, I want to say this: if you want to keep yourself updated, if you want to keep pace with the world, you must keep your knowledge of science updated. In order to do this, the best option is to subscribe this channel and keep connected with it. Hit the bell icon, and if you have any kind of questions, let me know in the comment box sections. And I must say that I found this video quite helpful. After telling you guys, after studying a lot, I literally got a lot of interest in the field of evolution and genetics, that I really want to pursue my career in this field, and I definitely want to meet Svantee Paabo in person. Thank you so much for hearing me, for listening me. I hope you definitely know this knowledge and try to implement in your daily life. Allah Hafiz.